Hey everyone. So we're gonna jump into your exam 3 review. I hope you all had a good spring break and are getting ready for these last few weeks of classes. I know time will go by super quickly, but I hope you had a good break and got to enjoy yourselves for a little bit. Before I start this review, I will not be on campus on Tuesday and Wednesday. I'll be traveling. So I will not be able to hold in-person office hours on Tuesday. So if anybody wants to meet in person or has any questions, um, I'll be free on Monday to hold in-person office hours or to go over some stuff with you in person. And I should still be available to hold office hours as usual over Zoom on Tuesday and Wednesday. But please email me so I know because I will be at a company site visit, so I won't be on my phone or computer as much. All right, so let's jump into your review. So the first section or first unit you covered was about biological reactions and enzymes. So we know that in nature, not all reactions are always favorable by themselves. Not every reaction is exothermic, for example. Oftentimes, we require energy input for reactions to happen, or some reactions are spontaneous, but they are really slow. So they may be thermodynamically favorable, but kinetically they aren't. So some of them may take hours, even days, weeks. Some reactions even take years to come to completion. In living systems, obviously, that's not what we want, and that would be really unfavorable for us. So we need to figure out a way to speed up reactions. So one thing that we can do is we can increase the temperature, which will increase the collision energy. So if you look at this graph, you can see that at normal temperature, the fraction of collision is not as high, but if we increase the temperature, the fraction of collision is a lot higher because we have more collision energy and the likelihood of two substrates colliding is a lot higher. We can also increase the concentration of substrates. So obviously, the more substrates we have present, the more likely they are to collide with each other and therefore react. And lastly, we can add a catalyst, which in nature or in living systems, catalysts are enzymes. So if we have an enzyme molecule and a substrate, they form an enzyme substrate complex, and the enzyme helps the substrates to react, and we get our final products. So enzymes are usually proteins and they participate in reactions, but they are not consumed, meaning even though they may actually change their conformation over the course of a reaction, at the end, they will be restored to their natural or to their previous state. So they will not be consumed in the reaction versus the substrate will be turned into a product and will not be the same as before. Because enzymes are so important, their activity is highly regulated in living systems, so they don't just randomly go about catalyzing reactions, but they are very tightly regulated. And enzymes have an active site where the substrate can fit into, and that is where the substrate will lastly react to form a product. So if this is our enzyme, we can see that the active site is somewhat the shape of the substrate, and we will get back to these two different types of theories in a second. But the substrate and active site fit, and the active site is specific to a substrate. And then they form that enzyme substrate complex, and at the end, we get our returned product. There are 7 different types of enzymes. They can be classified into 7 different classes, and they usually involve either the addition, removal, or rearrangement of certain groups. The enzymes are named after the class of reaction that they catalyze and the suffix ase at the end. So we have oxyreductase, transferase, hydrolase, lyase, isomerase, ligase, and translocase. So the first one was oxyreductase. As the name says, oxidation and reduction reactions are catalyzed by this class of enzymes, so the removal or addition of electrons from a substrate. For example, in this one, we have alcohol dehydrogenase, which oxidizes ethanol to acetaldehyde with the help of a cofactor NADH, and I will get back to cofactors in a little bit as well. Next, we have the class of transferases, also in the name, transfer functional groups from one molecule to another. So for example, we have hexokinase here, which transfers a phosphate group with the help of an ATP molecule. So we get glycerol, and we add a phosphate group, and we get glycerol phosphate. Other possible groups that can be transferred are amino groups, methyl groups, acyl groups, and so forth. Third, we have the class of hydrolases, which catalyze the hydrolytic cleavage of one molecule into two molecules. So we get an addition of water and molecule of H2O, and we cleave a bond. Next, we have lyase, which catalyzes the addition or removal of a functional group from or to a molecule with the rearrangement of electrons. So usually the formation of a double bond accompanies this type of reaction. For this, different sections use different examples, so I don't want to focus on one specific one here, because I'm sure that different sections will focus on different examples, but just understand the basic principle behind this type of reaction. Next, we have the class of isomerases. So isomerases catalyze rearrangements within a molecule. So functional groups are transferred within a molecule itself. So not between different molecules, but within a molecule itself. There are two different types of isomerases. One would be a mutase, which transfers a functional group from one place to another within the molecule. Or we have a epimerase, which invert functional groups about an asymmetric carbon. So if you remember this from the carbohydrate section, which we'll get to a little bit later in this review, we have chiral carbons, and functional groups can be on one side or the other, and an epimerase would transfer those functional groups or invert them from one side to the other. Next, we have ligase. This should be a familiar name to everyone if you think about DNA replication. So ligase joins two molecules to form one molecule with the help of ATP. So ATP hydrolysis is used as an energy source for this type of reaction. And lastly, we have translocases, which assist in moving molecules across a cell membrane. All right, so how do these catalytic mechanisms work, and what's the whole point of this? So to react, we know that two molecules must be physically close to each other, so they can't just react from one place in the room to the next just by looking at each other. They need to actually physically touch. And when those two molecules interact with each other, they form a transition state, which is the point of highest energy. And in this graph, we can see this right here. So we have free energy on the y-axis and the progress of the reaction on the x-axis. So the point of highest energy would be the transition state. And this is exactly the energy change that needs to be overcome for a reaction to occur. So it doesn't matter if the products have less energy than the reactants and the reaction itself is exothermic or thermodynamically favorable. We still have this little bump that we need to overcome for a reaction to happen, which is then called the activation energy. So the free energy of the transition state minus the free energy of the substrate. So this would be the activation energy that we need to overcome. And this is where enzymes come into play, because enzymes lower the activation energy. So they are catalysts, and they lower the amount of energy required for a reaction to occur. So as you can see in this graph, we have the free energy change of an uncatalyzed reaction versus a catalyzed reaction, and we can see that the activation energy is significantly lower. However, what's important to note is, again, the energy of the products and reactants is not changed with the help of a catalyst or an enzyme. The only thing that is changed is the amount of energy that is required to reach the transition state. Also, a lot of times, a cofactor is required for an enzyme to do its work, which is a small molecule that is required, for example, for the electron exchange or for the for ATP, for example, supplies energy for a reaction to occur. So we need different types of cofactors sometimes. Some important terms you guys should all know when it comes to enzymes and reactions. We have substrates, which is the substance upon which an enzyme acts, so the initial reactants. Next, we have products, which are the substances that come out at the end of a reaction. We have an active site within the enzyme, which is where the substrate binds on an enzyme. Again, we have cofactors, which are small organic molecules or metal ion that is required for the catalytic activity of an enzyme. Then we have the transition state, which is the unstable, very high energy intermediate that is formed in an enzymatic reaction. It usually has properties of both the substrate and the product. And then lastly, activation energy, which is kind of the key when it comes to enzymes, which is the threshold energy that is required to produce or progress in a chemical reaction. There are 3 different types of enzymatic catalysis, so 3 different types of reactions that occur within an enzyme. So we have acid-base catalysis, in which an enzyme transfers or helps transfer a proton. So a proton is transferred between the enzyme and the substrate. We have covalent catalysis, in which a covalent bond is formed between a substrate and a catalyst during the transition state. So usually there's a nucleophile in the enzyme active site, which forms a covalent intermediate with a substrate in search of an electron poor center. Usually in covalent catalysis, it's a 2-part reaction process, so the reaction graph has 2 bumps. So it creates 2 different transition states that flank an intermediate. And then lastly, we have metal ion catalysis, where metal ions mediate oxidation reduction reactions and promote the reactivity of other groups in the enzyme's active site through their electrostatic effects. Okay, so how do enzyme and substrate bind to each other? So we know that the active site residues are what's important for the reaction. So if you think about the 3D structure and how proteins are, like how they look in a 3D space, and how the amino acid residues in the active site interact with the substrate. So we have acid-base catalysis, covalent binding, metal ion catalysis. So the binding pocket has residues that can help position the substrate properly into the active site for it to participate properly in the reaction. We then form the transition state, which is the most unstable, and enzyme and substrate can form temporary hydrogen bonds to help stabilize that transition state, which is also why such high energy is required to reach that transition state. And the hydrogen bond length also determines the rate of the reaction. So if we have a short and strong hydrogen bond where the proton is equally shared between the donor and acceptor, it helps speed up the reaction rate in reactions. And again, reactant and enzyme must come together in the correct orientation in which the binding pocket residues help us. And as I mentioned before, there are 2 different types of theories of how enzyme and substrate come together. This is more of an outdated theory, the key and lock model, in which the key, which is the substrate, would fit perfectly into the enzyme. So the enzyme has a certain conformation, which is rigid and set, and only one substrate can fit into it like a key. However, that would mean that the active site fits exactly the shape of the substrate and that there's no alterations in the tertiary or quaternary structure of the enzyme. The more modern theory for how an enzyme and substrate bind would be the induced fit model, where we know that enzymes are not rigid, but they can kind of move about and the conformation can slightly change. They can adjust their shape. And as soon as the substrate comes into contact with the enzyme, it will kind of mold itself around the substrate to fit the substrate perfectly. So tertiary and quaternary structure are modified to fit the enzyme, the substrate. All right, in real life, an enzyme that is super important are serine proteases, which help leave bonds in proteins, so break down proteins. These are super important when it comes to, again, the breakdown of proteins. And the activity of serine proteases depends on 3 different active site residues that are also called catalytic triad. There are 3 different serine proteases: chymotrypsin, trypsin, and elastase, which actually have a very different amino acid sequence, but are structurally very similar, which is pretty interesting, but it shows that they carry out the same type of function. All these enzymes, all these serine proteases have a specificity pocket, which is a cavity on the enzyme surface that is close to the active site that helps accommodate the residue on the N-terminal side of the peptide bond that comes in. And the catalytic triad, so these 3 amino acid residues that sit in the active site are involved in hydrolyzing the reaction, or in the hydrolyzing reaction. So we know that these serine proteases are synthesized as inactive precursors, which are also called zymogens, and then are later activated. And this is really important because, again, they break down proteins. We do not want them to just go around breaking down random proteins within the body. We only want them to break down proteins where we need them to break down proteins. So they are synthesized as inactive precursors and then activated when and where we need them to. So enteropeptidase activates trypsinogen, which then the active form is trypsin. And trypsin, once it is active, can then act on the other zymogens, so chymotrypsin and elastates. And other enzymes can actually activate themselves, so autoactivation. So chymotrypsin is kind of a mix of those two. So chymotrypsinogen is the inactive form. We already activated trypsin, which then cleaves the inactive chymotrypsinogen, cuts out a certain bond, and then we get the active chymotrypsin, which then cleaves another bond within itself, and then we get the actual active form of chymotrypsin, which can then aid in the breakdown of proteins. And all of these happen in the small intestine, where the breakdown of proteins is actually needed. All right, jumping into the next section is enzyme kinetics. So how is the rate of reactions determined? So we have thermodynamics, which tells us whether a reaction is spontaneous or non-spontaneous. And we have kinetics, which tells us about the velocity of a reaction. So the change of a substrate or reactant concentration over the span of time. Looking at this in this graph, if you think of reaction velocity as concentration versus time, a substrate is used up. So we start with a lot of substrate, and as time goes by, the concentration gets less because we produce product at the same time. So these two are exactly related to each other. The more enzyme we have, the higher the reaction velocity, and we have a linear relationship between reaction velocity and enzyme concentration. To put this into terms of an equation, the change of substrate over time equals the change of product over time, but obviously inverted. However, what's important to note is that there is a nonlinear relationship between substrate concentration and velocity, and that is because substrates, the more substrate we add, only in the very beginning will the reaction velocity increase very rapidly, because at some point, all enzymes will be occupied with the substrate. So there's only a limited amount of enzymes that can be occupied by a substrate, and once all of these are saturated, the curve will kind of start to flatten out, and our reaction velocity will slow down and level out. So there is a maximum velocity that can be reached within a reaction. So if every enzyme has substrate present, then adding more substrate will not increase the rate. And this then takes us to reaction orders and reaction rates. So reaction rates is the rate equation, expresses the progress of a chemical reaction. The order of a reaction is determined experimentally and is expressed in terms of a rate constant and the substrate concentration. So we can calculate or determine the reaction rate of a reaction, so how fast a reaction will happen. There are three different reaction orders. We have a zero order reaction in which the rate is only determined by the rate constant. We then have unimolecular reactions, which are also first order reactions, and the rate is determined with the reaction constant as well as the concentration of a substrate. And lastly, we have bimolecular reactions in which the rate is dependent on the reaction constant, the rate constant, as well as both concentrations of two substrates. And if it's the same substrate, it would be that substrate squared, or it would be the concentration of one times the concentration of the other. Also note that the rate constant always has units that cancel out with the units of the concentrations. So therefore, you can also determine if you're using the right rate equation. This then transitions us to steady state kinetics and the assumption that the formation of product is a two-step process and that the enzyme substrate complex formation will be at equilibrium. So if we assume that we have a lot more substrate present than enzyme, we have enzyme concentration and substrate concentration, and those two form the enzyme substrate complex, which then reacts in turn back to enzyme and substrate. So not every enzyme substrate complex will remain an enzyme substrate complex, but it will form an equilibrium between forward and reverse reaction, as well as enzyme substrate complex forming enzyme and product. So if we take this assumption, we can assume that enzyme substrate complex concentration remains constant until all substrate has reacted, and therefore the change of enzyme substrate concentration over time is 0. Now, if we think about this in terms of reaction order, the first reaction will be a second order reaction because we have the concentrations of two reactants to take into account. So that would give us this reaction constant 1, which is a second order reaction. The reverse reaction is a first order reaction because we only have to take into account this enzyme substrate complex, which in itself is now a reactant. So K minus 1 would be a first order reaction. And the second reaction, the formation of product, is also a first order reaction because again, we only have one reactant. So then the change of concentration of enzyme substrate complex over time can be expressed with this rate equation, which if we express this in terms of only rate constants, we get this Michaelis-Menten constant. So this Michaelis-Menten constant gives us the substrate concentration at which the reaction velocity is half of the maximum velocity that can be reached. So this entire equation can be displayed graphically if we plot reaction velocity over the substrate concentration. So on the x-axis, we have substrate concentration, and on the y-axis, we have reaction velocity. And the Michaelis-Menten constant is plotted right here at V half max, 50% of V max. The substrate concentration can be read right here. Km can then be used to describe enzyme affinity for a substrate. So how likely or how much affinity an enzyme has to bind a substrate. The smaller Km is, the higher an enzyme's affinity for a substrate, because that means we require less substrate to reach half of reaction velocity, and vice versa. So if Km is really high, then the enzyme affinity is really low. This takes us to our next constant, which is the catalytic activity constant, Kcat, the rate constant of a reaction when the enzyme is fully saturated with substrate, which can be expressed in Vmax over the total enzyme concentrations. So essentially, this gives us the turnover number, or the number of substrate molecules that can be converted to product per enzyme molecule per unit of time. So again, I know this is a lot to take in, but Kcat essentially tells us how many substrate molecules can be converted to product per total enzyme that we have present. And then if we take all of this together, we can calculate the catalytic efficiency of an enzyme. So think of this as the likelihood of an enzyme and substrate colliding, the enzyme then binding that substrate, and then the enzyme converting that substrate to product. So this kind of takes all of this together and tells us how efficient an enzyme is in converting a substrate to product. The upper limit of catalytic efficiency, so the highest catalytic efficiency an enzyme can reach, would be the diffusion limit, because essentially this would mean that as soon as a substrate and an enzyme come together and build the enzyme substrate complex, it immediately gets converted into product. So that would be the fastest an enzyme could react. So now if we take all of this information, all of these constants, and plot them in a linear fashion, we get the Lineweaver-Burk plot. Again, this linearizes all the data we just talked about. So if we take the reciprocal of both sides, we can get a regular slope equation. So if you think of y equals mx plus b. So 1 over v would be 1 over the velocity, which would be y. The slope would be km over v max, and x is 1 over the substrate concentration, and then our b would be 1 over v max, which is the point where the graph cuts the y-axis. So all of this tells us, one, we can derive the maximum velocity as well as the Michaelis-Menten constant from a graph like this. However, it's not going to be as easy as all of this just was. Some enzymes do not operate at Michaelis-Menten kinetics. That's because some enzymes can have multiple substrates. They can have multiple steps or multiple active sites. So each of those enzymes or substrates with multiple substrate binding sites has a unique Km for each substrate, and Vmax is then reached when both substrates or when all substrates saturate the enzyme. So it's not going to be as easy to determine Km as it is in the other ones. All right, I've already mentioned that enzyme activity is very tightly regulated. So, for example, those enzymes can be inhibited. There are two different types of enzyme inhibition. There is irreversible and reversible inhibition. Irreversible inhibition, as the name already says, cannot be reversed. So this is final. As soon as the inhibitor binds to the enzyme, it can no longer be used. So an inhibitor binds to the active site instead of the substrate. It looks very similar to the substrate and usually forms a covalent bond that cannot be reversed and will block the substrate binding site forever, for good. This cannot be used anymore. There are also certain suicide inhibitors, which would be when an enzyme binds in a normal fashion to a substrate and starts its reaction, but then it gets stuck and cannot release the substrate anymore, and the enzyme will be unusable. We also have reversible inhibition. Contrary to irreversible inhibition, this can be reversed. There are 3 different types, and they all sound super similar, so this is really fun. We have competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. First, the competitive inhibition inhibitor and substrate will bind at the same active site. That's because the inhibitor is very similar to the substrate or to the transition state. Actually, it will be more useful if the inhibitor is more similar to the transition state, because that will bind more tightly to the enzyme. However, the inhibitor can be removed with the addition of more substrate. That's why it is called competitive inhibition. So inhibitor and substrate compete for the same binding site. If we plot this in terms of the Lineweaver-Burke plot, we get, sorry, my dog is grunting at me right now. We get an increase in the Michaelis-Menten constant because the enzyme has less affinity for the original substrate. However, the same Vmax will be reached. Duke, on my way. So if you look at this Lineweaver-Burke plot, we still have the same y-axis intersection. So the Vmax will still be the same. However, you can see that the slope has changed. So the slope is affected by the degree of inhibition, which can be calculated with the inhibitor concentration and the inhibitor constant. Next, we have uncompetitive inhibition, in which the substrate does not interfere with the enzyme, but it binds to a different side, or to the same side, but it doesn't interfere with the actual binding of substrate to enzyme. This slows down both Vmax and Km, so both the affinity of the enzyme to the substrate is altered, as well as the maximum velocity the enzyme can reach, just because the conversion of substrate to product will be inhibited or slowed down. And lastly, we have non-competitive inhibition, which affects only Vmax. That's because the inhibitor does not bind to the same side as the substrate, and does not alter substrate binding. So Km is not affected. So if you look at this Lineweaver-Burke plot, you can see that Vmax is affected. This is for, oh my God, I'm so sorry. My dog is biting my hands. So here, this is for the uncompetitive inhibition, in which both Vmax and Km are affected. So you can see that the y intersection is different, as well as the slope. And then there's also mixed inhibition, which can affect both Km and Vmax. So it can either increase Km or decrease Km. So it can go both ways, and it will decrease Vmax. So if you look at this Lineweaver-Burke plot for non-competitive and mixed inhibition, both Km will be affected. So it can increase or decrease, and Vmax is also affected. Okay, and we can also have allosteric interaction between certain substrates and an enzyme. So some enzymes have multiple binding sites to which certain molecules can bind, and they can either increase or decrease the affinity of an enzyme to a substrate. So it can be both inhibition or activation. This is really important, especially for feedback inhibition in certain metabolic pathways. So if you think about a certain pathway in which an enzyme regulates a very crucial step, if there's a lot of product produced at the very end, that certain product can act as an inhibitor for that enzyme and can then inhibit the enzyme as long as that product does not need to be produced anymore. And when that enzyme is needed again because product got depleted, that product will not bind to the enzyme anymore, it won't inhibit it anymore, and the enzyme can be used as before. All right, jumping into the next section, carbohydrates. So carbohydrates are the most abundant biological molecule. We also know them as sugars or saccharides, and they have a variety of cellular functions. Usually that would be energy storage, structure, communication, or they can be used as other precursors for biological molecules. So usually a carbohydrate would be some sort of carbon molecule that reacts with H2O to form a carbohydrate. And the structure of a regular carbohydrate would be CH2O, and that is a repeating unit of at least 3 of those. So carbohydrates can be classified by the number of carbons they contain. Like I said, the minimum number would be 3, so that would be a triose. We can then get a pentose, hexose, heptose, and so forth. Hexoses and pentoses are the most abundant in living cells, just because they are the most stable, and they can form this beautiful ring structure right here. So this and this is the same exact molecule, just in a different arrangement. There are also certain carbohydrate derivatives, many of which include certain groups containing nitrogen, phosphorus, or other elements. And all carbohydrates are easy to recognize by the large number of hydroxyl or OH groups. All carbohydrates contain a carbonyl group, either in the form of a aldehyde or ketone. So depending on where that carbonyl group is found, if it is found at the terminal end, it will be a CHO group, or if it is found in the middle of the carbohydrate, it will have 2 R groups and a carbonyl bond. So depending on if it is an aldehyde or a ketone, it will have aldo or keto as a prefix, then the number of carbons, and the suffix -ose. So for example, a ketohexose would be fructose, or aldohexose, which would be glucose. Each carbohydrate can have a stereoisomer, so these molecules differ from each other in their 3-dimensional arrangement, but they will still have the same molecular formula. This is because carbons that carry 4 different substituents are chiral, which means they have a certain spatial arrangement. So important, it has to be 4 different substituents. If it's 2 H's or 2 OHs, it is not a chiral carbon. It has to be 4 different substituents. So chirality then gives rise to enantiomers, So they have the same formula, but different stereoisomeric structure. We can have a D enantiomer where the OH is on the right of the highest numbered chiral carbon, or we can have an L configuration where the OH is on the left of the highest numbered chiral carbon. To identify the forms, to identify the different forms, we need to number from the carbonyl carbon. So this would be our carbon 1, carbonyl carbon, and then we count down to the highest carbon and determine the position of OH. There are also stereoisomers that are called diastereomers. Those are non-mirror images, enantiomers are mirror images. So diastereomers can be either epimers, where there's only one different chiral carbon, or anomers. Those differ at the anomeric carbon. Okay, like I already mentioned in the introduction to this, sugars can form cyclic structures. So if a sugar has 4 or more carbons, they will exist primarily in their cyclic form. 5-membered rings are called furanoses, and 6-membered rings are called pyranoses. So these ring structures are formed through intermolecular rearrangements, where the carbonyl group right here reacts with a hydroxyl group at the end and forms this bond right here. So carbonyl carbon OH group, and we get a cyclic structure. There are 2 different arrangements those rings can take on. They can either take on the alpha form, in which the OH group of the anomeric carbon will be in the opposite rearrangement as the CH2OH group, or the beta anomer, in which the OH group will be in the same configuration as the CH2OH group. Also, this is called Fischer projection, so that stick form right here, and the cyclic structure is called Haworth projection. I already mentioned that we have aldoses and ketoses, and depending on which one we have, the cyclization will look slightly different. So the carbonyl group will sit at different positions, and therefore how we form that ring will be slightly different. Right here, we have an aldehyde, and that will react with that C1 carbon, and we will not have a substituent on the anomeric carbon. This right here is a beta anomer, because look at this OH group, it is in the same orientation as this CH2OH. Right here, we have a ketose. The carbonyl group is on the C2 carbon, and that will react with the C5 carbon, which means our anomeric carbon will have a substituent. It will have this C1 carbon sticking out, and this right here is in the alpha form, because the OH group is pointing down in the opposite direction as that CH2OH on the C5 carbon. We have also seen different conformations or different displays of our glucose that is cyclized in cyclic form. This would be the chair conformation, and this is the form that usually glucose takes on because it is most stable. So the sugar is puckered, that's what it's called, in which the carbon has tetrahedral bonding arrangements, so the substituents are either above or below the ring. And this is especially then beneficial when the carbon carry bulky substituents, because they will point away from each other and will not get in the way with each other. So this minimizes repulsion between the groups. And this is also why glucose has such a stable form and is the most commonly found sugar in all living organisms. Because in glucose, all substituents are in the equatorial position, versus in the actual position in which they have more repulsion to each other. So we already know that we can have either alpha or beta arrangements of or ring structures, but in an aqueous environment, the alpha and beta form can readily interconvert between each other, as well as also between a pyranose and furanose ring. So they can either form a 4 or 5-membered ring, and all of these forms can interchange in an aqueous environment. We also know that we have different derivatives of monosaccharides. In this course, we've previously seen phosphorylated monosaccharides. So glucose, for example, can be phosphorylated. We also know that nucleotides in nucleic acids carry a sugar ring. So there are different derivatives of monosaccharides. And then one last important thing to know about these carbohydrates is that the anomeric carbon, so that carbon that carries the carbonyl group, if it is not participating in a glycosidic bond or does not carry any other substituents or bonds, it has a free OH group right here, then it is a reducing sugar. So it can partake in a reduction reaction and donate an electron. However, if this anomeric carbon carrying the OH group is partaking in some sort of glycosidic bond and is not carrying a free OH group, then it is not a reducing sugar. It is non-reducing. So this then takes us to the polymer of the monosaccharides, which are called polysaccharides. Makes sense. We have multiple of them, of these monomeric subunits. So carbohydrate chains are called glycans, and a glycosidic bond can be formed via a condensation reaction in which the free OH group on the anomeric carbon reacts with another OH group on another sugar molecule. So examples for example, disaccharides, so molecules that have 2 sugar subunits, would be lactose or sucrose. So in this example, we have sucrose, and this bond is named after the form that the first sugar has. So it is a alpha bond or a alpha anomeric carbon, because the OH group was pointing down. It is forming on the number 1 carbon, and then the bond on the other molecule is forming on the second carbon. So it would be an alpha 1,2 glycosidic bond. Since there are so many OH groups in a different sugar or in different sugar molecules, we can get very different types of bonding and branching in our polysaccharides. Okay, and this will then take us to the storage forms of glucose. So we have glycogen and starch, which are used to store energy in form of glucose monomers or glucose polymers in this case. So the polymers of glucose are linked via an alpha 1,4 glycosidic bond. So again, we have our alpha anomeric carbon, which bonds from the C1 carbon to the C4 carbon. So it is a alpha 1,4 bond. And we have hundreds of those subunits connected to each other via the same bond, and we have a huge polymeric chain of that. We can then get highly branched structures. So those super long chains of glucose polymers are linked via a 1,6 bond to each other. So we still have these 1,4 chains, and then they can also be connected to each other on the C6 carbon, so right here, and they can form longer branches to each other. So these branches are really important for the quick breakdown of glucose monomers and to make energy readily available. In animals, every 12 carbons on such a long polymer chain will carry a branch, and in plants, every 24 to 30 will carry a branch. And then another form of polymer that is made up of glucose monomer is cellulose, which is a structural polysaccharide. It is connected via beta 1,4 linkages, and it is a non-branched structure. It is the main component of certain cell walls and vegetable fibers, and it forms long extended fibers that give rigidity and strength. So that is because they are just super duper long and are not branched. Similar structures as cellulose can also be found in exoskeletons. So those are also strength giving or form giving. For example, if you think about chitin in insects, fungi, or crustaceans. All right, and then you guys should remember this name glycoproteins from our last unit. So those would be proteins that carry carbohydrate attachments. Those can be linked via either nitrogen or oxygen linkages. So we can either have an N-linked oligosaccharide or an O-linked oligosaccharide to an amino acid chain. So depending on which amino acid it is attached to, it is either an N or an O-linked glycoprotein. So if we have an N-linked protein, it is linked via asparagine, which is linked via glycosidases, and those glycosidases can attach entire carbohydrate chains, and then they will trim the chains depending on the amino acid sequence and structure that it is attached to. All of this happens while the protein is synthesized on the ER, and once it is synthesized, it immediately, or while it is synthesized, it immediately will get its carbohydrate attachment. In O-linked oligosaccharides or glycoproteins, those can be linked via either serine or threonine, and the residues are added one at a time, and this occurs in the Golgi apparatus and is carried out by a glycosyltransferase. These glycoproteins or carbohydrate attachments are really important if you think about, for example, blood groups. So oligosaccharides are added and then determine which blood group we carry. So that's then important when it comes to antibodies and so forth. And then lastly, we have proteoglycans and peptidoglycans. Proteoglycans contain repeating units of chondroate sulfide and are structurally looking like this. And bacterial walls are made up of peptidoglycans right here, which is a network of cross-linked carbohydrate chains and peptides. For this, just know where these different types of glycoproteins are found and how they look and how they are linked. All right, and then the last unit that was covered for this exam was metabolism and different cellular pathways. So metabolism is just a term that pretty much condenses all decorative and biosynthetic cellular reactions. So we have anabolism, in which, if you think about A stands for assemble, we go from smaller building blocks and we make larger macromolecules. For this, we require energy input because we are building things. And then we have catabolism, in which we break down these macromolecules into building blocks and we get free energy out of those reactions. And this is all connected in a bigger pathway, or these pathways are all interconnected between breakdown and assembly. So how do we get to all of our nutrients or all of our building blocks from the food that we take in? So all the food that we consume serves as a source of metabolic fuel or energy, and it gives us all the building materials we require for our cellular pathways. So we consume food to gain building blocks for all our cellular functions and to then catabolize them into different biomolecules. When we break down carbohydrates into monosaccharides, we break down proteins into amino acids, nucleic acids into nucleotides, and lipids into fatty acids. All of this happens during digestion. So as we take in food, we digest it and break it down. And for the breakdown of food, we need help of different enzymes. So this is where all of what we just learned comes back together. And all of this happens very quickly in our body, which is why we need those enzymes to help speed up reactions. So in our bodies, for example, we break down carbohydrates in our mouth. As soon as we smell food or see food, the enzyme production happens and all these enzymes are released into our mouth, for example, or into our intestine. So that's why smells are super important when it comes to food. So all these enzymes are released. For example, in our salivary glands, we have amylase, which breaks down a polysaccharide in our mouth into smaller molecules. And then those smaller polysaccharides then travel to the stomach, and with the help of pancreatic fluid, it gets broken down into disaccharides. And then in the small intestine, it gets broken down into monosaccharides. Lipids are broken down into fatty acids with the help of lipases, and proteins are broken down with the help of proteases. And I already mentioned earlier, for example, trypsin, chymotrypsin, and elastase. So those are super important proteases, and they are only active in the small intestine. And we already talked about inactive precursors and how they then get activated and why that is important. So nutrient breakdown, I already mentioned amylase that gets released in our salivary glands, hydrolyzes starchy foods into glucose residues. This happens in our saliva. So we have these polysaccharide chains and they get hydrolyzed. So with the help of water, H2O, we break these down into smaller polysaccharide units. Proteases leave peptide bonds to break down amino acid residues. This is only found in the stomach and small intestine. So we break this peptide bond with the help of a water molecule and get smaller polypeptides. And then lastly, fatty acids are released with the help of lipases that break down larger lipids into the fatty acid chains and glycerols. These fatty acids can then form micelles because if we remember the hydrophobic effect, we don't want hydrophobic molecules touching polar molecules or hydrophilic molecules, so they will try to avoid any aqueous surface and form a micelle. The broken down nutrients are then absorbed by cells in the intestinal lining. Saccharides are transported through active transport, amino acids via symporters, and fatty acids can be transported in a variety of different ways. Once the fatty acids are in the intestinal cells, they can either be cholesterol. So if you remember cholesterol, I think in the first unit, that is part of the lipid bilayer and an important transport protein, or they can also reform to triacylglycerols again. Adipocytes are specialized cells for the storage of lipids. So those are our fat cells that we accumulate, and they store lipids in form of triacylglycerols. We also know that glycogen is the storage form for glucose monomers. We just talked about this. So they can either be stored in the liver or muscle as medium-term energy storage in form of glycogen, or we can use glucose immediately for energy. And then amino acids, those are actually not stored, but are immediately used as building blocks for other polypeptides. So if we think about proteins or enzymes. So all of these nutrients are metabolic fuels and can either be broken down for energy purposes, like immediately using them to build other things or to require energy, so to drive cellular processes, or we can store the nutrients in a certain way so that the body can utilize it once it runs out of immediate fuel. All right, so how do we then break down our storage molecules if we do decide to store them? Glycogen is broken down via the addition of a phosphate group, so we need to expend energy to break down our energy storage, which is a little counterproductive, but at the end of the day, we get a lot more energy from a glucose molecule breakdown than we expend on breaking these glycogen chains. So we add a phosphate group, which is a phosphorylases, and we get an activated glucose 1-phosphate molecule, which can then enter glycolysis. Triacylglycerols are broken down via lipases in the same manner as they are during digestion, and they are broken down into fatty acids and glycerol, and they can then enter circulation and can be used for energy. However, these triacylglycerols are only broken down when other fuels are not available. Proteins can be broken down in 2 different pathways. We can either break them down in the lysosome or via the proteasome. And the proteasome pathway utilizes a degron tag, which is a tag that can be found in the amino acid chain. Proteins containing that tag get ubiquitinated, so they get a ubiquitin tag attached to it. And if there are 4 ubiquitin tags attached to a protein, it will be then broken down in the proteasome. So we have a protein that contains the polyubiquitin signal, and this gets then transported to the proteasome, and with the expenditure of ATP, the proteasome containing proteases will then cleave the protein into its amino acid subunits. All right. So what do we then do with all these subunits? All these different nutrients get broken down into its subunits and then can enter different metabolic pathways. So metabolic pathways are a series of reactions that are required to break down those monomeric units or to build them from smaller pieces. A lot of metabolic pathways share some common intermediates. So in glycolysis, for example, we split glucose into 2-pyruvate via this glyceraldehyde 3-phosphate intermediate. We then get pyruvate. Pyruvate is then converted to acetyl-CoA, which is then oxidized to CO2. As you can see, there are a lot of common intermediates that come from different pathways. So right here, alanine can be converted to pyruvate. We can also have oxaloacetate also converted to pyruvate, which can then also be converted to different amino acids, or we can enter the rest of the oxidative phosphorylation, the citric acid cycle, break it down to carbon dioxide. So there are a lot of common intermediates. Most metabolic pathways include oxidation reduction reactions. Catabolism usually involves oxidizing carbon, while the synthesis involves carbon reduction. Remember that oxidation is the loss of electrons and reduction is the gain of electrons. So if you think about this, or an easy way to remember this is that the reduction, we reduce the oxidation number because we gain electrons. In fatty acids and carbohydrates, carbons are oxidized to CO2 and energy. And electron transfer usually involves some sort of hydrogen and other cofactors. So electrons are usually transferred to a compound such as NAD+. If you think about oxidative phosphorylation, those cofactors get then recycled restored to its original form, and we produce energy in the form of ATP. So we know that all metabolic pathways are somehow connected. Again, remember all these intermediates, and pathway activity is highly regulated. Enzymes carrying out all those different reactions and all those different steps are usually regulated via feedback inhibition. We already talked about feedback inhibition, how a certain product at the end of a pathway can regulate an enzyme earlier on in the pathway. That's super important, so we don't expend unnecessary energy. Also know that not every cell carries out every pathway, only in those cells where it's necessary. So certain cells have necessary specialized functions. Each cell has a unique metabolic repertoire, so each of these carries different metabolites. And some organisms can be metabolically interdependent. So if you think about it, we depend on plants to do photosynthesis. Sorry. All right. And now we get to more math. Again, pathways are often linked, and we already talked about free energy changes, spontaneous and non-spontaneous reactions. Not all reactions are spontaneous, and we want reactions to happen regardless. So we can do that by linking those reactions together and use energy from favorable reactions where energy is produced to drive other unfavorable reactions where energy is expended. So remember that a delta G less than 0 means a spontaneous reaction and exergonic, and a delta G more than 0 is a non-spontaneous endogonic reaction. So all reactions with a net delta G of less than 0 will be spontaneous. So if we couple a reaction of a large negative delta G with a reaction that has a slight positive delta G, the overall delta G will still be negative and the overall reaction will happen. The free energy change depends on the reaction concentration, the reactant concentrations, sorry. We have our equilibrium constant, KEQ, which gives us, or which is calculated with the concentrations of the reactants and products at equilibrium. So if you remember, a reaction has a forward and reverse reaction, and at some point will establish an equilibrium between the forward and the reverse reaction. And then if we take those concentrations at equilibrium, we get our equilibrium constant, KEQ. And with this constant, we can then calculate the free energy change. This equation should look somewhat familiar if you remember from the last exam. Remember that all of these reactions depend on the biochemical standard state because we are in a living system. So the standard free energy change is happening at the same reaction conditions. If we want to calculate delta G and not standard delta G, we will use this formula right here. You guys should remember all this from your exams. Make sure you know how to use all these different equations, when to use them, which numbers to plug in. So definitely practice plugging in and solving for different variables, solving for example, KEQ. So this and this is the same. This is KEQ. So know how to solve for this, know which variable stands for what. Definitely understand how to use this. That will be important. All right. And then lastly, kind of circling back to the coupled processes, ATP is involved in pretty much all coupled processes because it produces so much free energy. So this right here is our ATP molecule. we have our sugar ring, our phosphate group, and our base attached to it. And we already know that we can make unfavorable processes happen by coupling them to other reactions with a large net free energy. Again, the hydrolysis of ATP releases a huge amount of free energy. So if we pair the breakdown of ATP, so the hydrolysis of ATP, which releases about 30 kilojoules per mole, with the, for example, activation of glucose to get glucose 6-phosphate, which would cost us 13 kilojoules per mole, we would get a net free energy change of negative 16 kilojoules per mole. So overall, that would still be a favorable reaction. And ATP is therefore so important in all of these reactions because it connects anabolic and catabolic pathways. So why does ATP release so much free energy? If you look at this molecule, ATP hydrolysis products will be much more stable than the reactants. That is because all of these phosphate groups are highly negatively charged, and all of these charges repel each other. So if we cleave all of these phosphates, we get much more stable intermediates. Other molecules can also serve as energy currency. Usually those involve some sort of phosphate hydrolysis. So usually they have some sort of phosphate bond attached to it. And the cleavage of that phosphate bond will then release a large amount of energy. The regulation of all cellular pathways usually occurs at steps with the largest free energy changes because we don't want to expend unnecessary energy or release energy if we don't need to. So we try to regulate all processes at those points where large free energy changes take place. All right, I think this summarizes most of what you covered in class. I hope this summary helped, and I hope that everyone will be super well prepared for their exam next week. If you have any questions, please feel free to reach out to me. I will try to answer your question as much or as good as I can, and I'm totally happy to meet with you tomorrow. So let me know if you do want to meet. We can meet somewhere on campus or we can meet on Zoom. So just shoot me an email and good luck.